So thank you very much for that, and of course, thanks for the opportunity to be here. I do want today, first I have to show I have my medical credentials here, the truth in advertising. I am a consultant for a company called Ionis Pharmaceuticals, who has been an intimate partner in part of the work I'm going to describe today. But the beginnings of the story that I'm going to tell is the recognition from human genetics that where we know genes that are either causative or contributory to disease, they're expressed broadly in many cells of the nervous system, not uniquely within individual kinds of neurons whose degeneration or dysfunction causes a specific disease. And even more so, work from an earlier Breakthrough Prize winner, John Hardy. He discovered that if you know how you inherit one copy of each gene from mom and one from dad, if you actually happen to inherit 3 copies instead of 2, perfectly normal gene, but 3 copies, and it's a specific gene encoding the product alpha-synuclein, you will get Parkinson's disease. And if you inherit 3 perfectly normal copies of a different gene, the one encoding amyloid precursor protein, you will get early onset Alzheimer's disease. So gene dosage matters. And no mutations needed there. Gene dosage matters. And you could also then recognize that, well, you could be directly on the mechanism causing those diseases if you could just lower the synthesis of the disease-causing gene. And we set out to try to do that, but started an example of the disease amyotrophic lateral sclerosis, or ALS. The Americans call it familiarly Lou Gehrig's disease. And this is a disease where the neurons, the upper and motor neurons, degenerate and then fail to function. The paths of these neurons were known and drawn here by the greatest neurobiologist of all time, Ramon y Cajal, there more than a century ago, where the upper motor neurons send their processes down the spinal cord and to connect to the lower motor neurons, whose job it is to innervate the muscles and to signal them to contract. The features of this disease are that it's called a late onset disease, although at my age is not, that's not, mid-50s is not so late, but it's the symptoms is a fatal paralysis over a handful of years. And for the students here, your lifetime risk is about 1 in 2,000 dying from this disease, and for the faculty, that's risen to about 1 in 1,000. And there's no really effective therapy to slow the degeneration of motor neurons today, but the salient feature that led to the beginnings of the molecular era of discovery was that about 10% of instances of disease are clearly caused in an inherited form when you inherit one damaged gene from one of the parents. And while 1 in 1,000 or 1 to 2,000 is not a really big number, if you do the calculations biologists can do, you saw those fancy physicists this morning, the biologists, that means there are about 4 million people now alive who will die from this disease. Okay, so, and here's the founding report in the journal Nature describing mutations in the first gene whose mutation is causative of ALS. And it's a gene that encodes a protein which is expressed in every cell in your body. And yet, later in life, if you have a mutation in it, you're going to develop this fatal paralytic disease. And when we got started, it was thought that the damaged gene just didn't do what it's supposed to do. But we demonstrated that actually, no, it's not that it doesn't do what it's supposed to do. It's that it does something that it's not supposed to do. It does something bad. And it's a little sobering in the scientific community that actually 24 years after the original discovery, we agree that it does something bad, but we don't agree what the bad thing is. And there are 9 prominent models. Additionally, we demonstrated that actually it does the bad thing not just in those motor neurons, the ones that are going to trigger the muscles to contract. It does it by damaging the cells that surround the motor neurons and naturally nourish them. And that the disease is really a disease of the system, with damage within the motor neurons really amplified by damage by the neighboring cells. And so then again, so a strategy there for then combating disease would be, can you just silence the bad gene in one or more of the cell types whose synthesis of the bad gene product is really driving disease? So we set out to do that, and the way we're going to do this is to take little designer DNA drugs. The genome is made up of genes, the genes are made up of DNA, has a 4-letter alphabet, and we're going to make a designer DNA drug that matches the first intermediate information copy made from the gene. It's called an RNA. And if it completely matches and is self-complementary to it, you make this hybrid helical molecule here, half DNA, half RNA. And that makes the RNA, this information intermediate, it makes it a substrate for an endogenous intranuclear enzyme that destroys the RNA. It sort of looks like a viral intermediate, and it destroys the RNA. So if you do that, you shut off the flow of information from the gene to its final product, the protein. And over the last 20 years, we're sort of in version 2.5. I am in Silicon Valley. So you're in version 2.5 of the medicinal chemistry modifications to the underlying designer DNA. And you'll see what happens now when you incorporate those changes into these molecules. And here's the founding experiment. And it was to ask, can we lower the level of the target RNA encoding this gene? It's called superoxide dismutase. And here's the level in normal or mock-treated animals. And you can lower it near the site of infusing into the nervous system one of these designer DNA drugs. You can lower it. Oh, actually, you can lower it all the way throughout the entirety of the nervous system. And if you do that, and you take an animal that is a genetic mimic of the human ALS disease, the animal expresses this ALS-causing mutation. What you see is that those animals only live 25 days after disease onset, but if you treat them once with this drug, you can double survival after onset. And so that was the founding experiment that maybe these designer DNA drugs in the nervous system really could work. And here's the founding paper, now more than a decade old. Okay, so, and here's the scorecard of where this approach now is in nervous system disease. We know that we can deliver the drugs broadly in the nervous system. We can alter the expression of target genes. And an amazing feature you'll see in a moment, you really get, you take an aspirin, you get 3 or 4 hours of relief. Well, here we don't get 3 or 4 hours. We don't get 3 or 4 days. We don't get 3 or 4 weeks. We get 3 or 4 months from single dosing. And it's regulatable, it's commercially feasible today, and it's been demonstrated to be safe in 2 applications. There are disease applications in this disease ALS. In a disease example, I'm going to show you where we are in a moment in a different disease. And there's an approval in one of the abundant genetic diseases of children called spinal muscular atrophy that was approved last December. Okay, so that's where we are. But one of the donors of the Breakthrough Prize, Yuri Milner, has given us a charge. Tell us what the future is going to look like. And so here it is. So there are many diseases for which this gene silencing approach would be appropriate. I've just said, I've shown you the first example in ALS, but the largest cause of ALS is mutation in a different gene, which also does something bad. It's the largest cause of ALS and the largest cause of the second most frequent dementia, and lowering that would be good. You could target another nervous system disease, the ataxias, again, where there's a damaged product. And you could look, and indeed, from work from Stanford, from Aaron Gittler and his colleagues, has demonstrated that one of those ataxin genes is probably the best target today for sporadic ALS. And a previous Breakthrough Prize winner has studied a gene, MECP2, which is involved in inherited intellectual disability, and the approach will be going forward in that disease. And I'm going to show you an example of chronic brain injury. I'm sure you've heard about it, a National Football League disease, and how we could approach that. And even the big diseases, lowering that gene alpha-synuclein that I introduced at the beginning, doing that could be therapeutic. And then there's Alzheimer's disease, the biggest disease of all, lowering amyloid precursor protein. And lastly, I am in the Ludwig Institute for Cancer Research, and indeed the fatal cancer of the nervous system, glioblastoma, targeting the genes that allow rapid growth, uncontrolled growth of those cells, we could do that. Okay, so let me just now go back to where are we? And there's Huntington's disease. Well, okay, so this is a disease about the same number of affected individuals as ALS. It's 100% genetic from the same gene. And the gene does something bad broadly in the nervous system. We know it does something bad, and just like the ALS example, the medical community, we don't agree on what the bad thing is. But what we showed was that if we take animals that develop Huntington's-like disease and treat them in a single-dose therapy, we can get disease reversal. Well, partial disease reversal. But that partial disease reversal with a single-dose therapy lasts a long time. And here's such an example of that. You're injecting the drug into the nervous system and looking at the efficacy. You can see you lower the gene product right after injection of the drug, but even 8 weeks later, it has not come back to the original level. So we are getting sustained efficacy by single-dose injection. And when we did that, and that went to human trial in the summer of 2015, and the outcome of that trial is to be reported next month. I'm not involved directly in the clinical approaches. We're the team underneath that, and I'm a cheerleader, and you can probably tell that. I'm a cheerleader, so we're hoping that we've actually done good. Okay, so then in 2011, the most frequent cause of ALS was discovered. It turns out it's actually the most frequent cause of the second most common dementia as well. Same gene, mutation gives rise to 2 different diseases. And it was found by others on September 21st. We designed the therapy on September 22nd. And we achieved proof of principle in July of 2015, and we're hoping that we'll get to trial by the beginning in the first quarter of next year. And then, now here's the founding case. It was mentioned in my introduction. This is one of my first discoveries. It's now 40 years ago today. And it was the discovery of a protein named tau, which we now recognize to misaccumulate inside the cells that are affected in Alzheimer's disease. And indeed, not only does it accumulate aberrantly inside those cells, both in Alzheimer's disease and chronic brain injury, but it's the intracellular tau tangles are the best correlate of cognitive decline. And so the strategy now will be to target tau, lower the level of synthesis of this gene, a normal gene, but to lower the level of its synthesis. And indeed, Ionis and the big pharma Biogen took that to trial just last month for an attempt at therapy in Alzheimer's disease. And then one more example here. I'm sure all of you are familiar with this chronic brain injury, or sometimes called National Football League disease. And there are prominent players who have been affected. And here, the diagnosis of the disease at autopsy is these little misfolded tau, this protein that I described before, misfolded tau. And if you look at blow-ups of that, it's just like it misfolds inside one neuron, then kills that neuron, slowly spreads, slow cell-to-cell spread. And Peter Walter this morning described one approach to try to suppress it, and here's another to try to suppress tau. And now, 2 last examples of the future. And can we not just turn the gene down, but can we turn the gene off? Can we do gene editing? 2 previous Breakthrough Prize winners have pioneered the technology to be able to edit genes in cells. And now we want to do that throughout the nervous system. And our strategy is to express the nuclease. This is a nuclease that can be targeted to individual sequences, but it's targeted by a specialized, unique sequence RNA. Oh, that's the little designer DNA molecules I was talking about before. Actually, they've been modified, so they're not really RNA, they're not really DNA. They're a little bit hard to tell. And anyway, so we're going to transiently activate these scissors, these gene scissors. We're going to transiently activate them by infusing one of these antisense oligonucleotides. That will then disrupt the gene that we're trying to target, but then the scissors will turn off with the natural decay of the designer DNA drug that we add. And so you don't have scissors just running all the time. You can just turn them on transiently, which we think is likely to be a requirement for really doing this. And then lastly, the most out there approach that I'm going to describe today, it's that in these diseases of the nervous system where neurons degenerate and die, you probably have heard that there are many efforts, especially in California, to replace those neurons using stem cell approaches. We think that we can jump that by now converting. One of the founding Breakthrough Prize winners just showed us that if you took a cell that had already become a specific cell type and you could actually convert it back to an embryo-like state, and subsequently, we've realized, especially one of my colleagues in San Diego has realized that we can convert them directly from one cell identity into a neuron. And so here's the approach. We're going to take our designer DNA drugs to reset the cell identity, converting glial cells, of which there are a huge abundance in the nervous system, directly into neurons. And so we think we can, by altering expression of selected genes using this DNA drug approach, that we can replace the neurons with new neurons that were converted from non-neurons. And have we done that today? Yes. Oh, in cell cultures. Have we done that in animals? Not quite. Do I think it's going to work? Yes, I do. Anyway, so that's where we are. And let me just now close by describing the team. These are the key team players. There were a trio of us at UC San Diego, Tim Miller, Richard Smith, who drove this effort, the chief scientific officer of Ionis Pharmaceuticals, who was a key partner all along through the initiating work. The work on Huntington's disease was really spearheaded by a single postdoc, Holly Cortesewicz, and the work on the most frequent cause of ALS was partnered with Clotilde Lagier-Tourien, now at Mass General Hospital. And the work on spinal muscular atrophy is not my work. It's the work of Adrian Craner at Cold Spring Harbor. And I should mention that this was funded at the beginning almost exclusively philanthropically by the ALS Association and by the long-term support I get from the Ludwig Institute for Cancer Research. Thank you very much.
Okay, we have time for a couple of questions. Yeah, here on
There's two back there.
Thank you. Had you considered using low-level laser therapy for neurogenesis to create new brain cells rather than converting glial cells?
So have I considered that? Will there be multiple approaches to be able to generate new neurons? I certainly hope so. In our case, we think that that the approach is actually very straightforward and quite simple to do it by this designer DNA drug approach. But our charge up here was to try to describe what the future might be. And I'm sure we're going to be, with a big W, we're going to be able to do that at some point. I think the strategy I outlined has a real chance. But others, I would, if others achieve that, I would just sit back and applaud. Yeah.
More questions?
Yeah, there's one right here. There's another one back there.
Thank you for your talk. In order to function properly, neurons need glial cells around them. So if you convert the glial cells into neurons, what's going to be converted into glial cells?
Oh, you're certainly right that the neurons completely require these glial cells for a variety of support mechanisms. We certainly don't intend to convert. There are more glia than there are neurons. We certainly don't intend to convert all of them. But the glia are reborn, so we expect that the sources, the glial stem cells already there, will now repopulate the glial population. We're going to only aim at borrowing a few percent of the original glia. Of course, that's a dosing effect, and that's, in part, I would argue, that's the beauty of the designer DNA drug approach. We have dosing capabilities to try to determine that. Yeah.
There's a question here in the center.
Yes, thank you. My wife, several years ago, a certified nutrition consultant, published a case study in a journal suggesting that cysteine and sulfite toxicity is a primary cause of ALS, that people who, if you can completely get rid of the sulfite in a person's body, their nerves won't get any worse.
Yeah.
And she was able to stabilize one patient. So I wonder if people like Stephen Hawking, who've lived for years with ALS, have something that prevents sulfite from eating up their nerves.
Yeah, so I think we don't know that at all. Stephen Hawking absolutely positively has a motor neuron disease. It's been very slowly progressive, well outside the typical norm for the disease ALS, and so it's a very unusual case. So I'm not sure we can use Hawking as a test case for general disease. Yeah.
Okay, there's a question in the back on the far left.
Would you be kind enough to update us about any immunotherapy modalities these days to potentiate body's own immune system for sort of diseases where you have misfolding proteins, as you mentioned, Alzheimer's disease?
Okay, so an update on immunotherapies in the nervous system. So the most prominent one by far have been a set of trials in using antibodies to try to clear the extracellular deposits called A-beta in Alzheimer's disease. There have been a couple of spectacular failures, one about a year ago from Lilly, and there are several now ongoing, one from Biogen, one from Genentech, using alternate antibodies where I think there's high optimism in many circles, but we haven't really demonstrated yet whether any of them can actually work. I'm actually an enthusiast about parts of that, but I'm also an enthusiast that I really doubt for these major diseases that a single therapy is going to fit all. And so I think that, again, I would applaud those efforts, but I'd also say other approaches should be ongoing in parallel, both the ones I described and others.
Okay, we have time for one quick last question.
Is there something specific about neurological diseases that make them better targets for this antisense oligo approach?
Yes, I do. I think the selectivity for why these are so good in the nervous system is because we get these long-term efficacies. Almost no cells are dividing. So if you take up these drugs, then you now have them for long, long periods. And the chemists have now modified them so the DNA drug really has no natural enemies inside the cell, and the drug just sits there for weeks and weeks and weeks, catalyzing further RNA decay.
Okay, let's thank Dr. Cleveland again.